So let's get going on this Ubiquity of Microorganisms lab. So first things first, you always want to make sure that you have everything you need. So I have the 4 TSA plates that were prepared. I have distilled water that I just poured into this cup, the incubation chamber, the thermometer. I've got 2 sets of swabs, sterile swabs. Each one has 2, so that's 4 swabs. I've got my fire in a can. I've got my inoculating loop, something to hold on to it, isopropanol, and my lighter. And so the other thing is our instructions. And so what I've loaded up on the website is a PDF of the instructions that I took directly from the Hands-On Lab website. But what I did was I also made sure that, and you'll see this in the website, I annotated things. So you'll see here it says instructor notes and then Willette addition. Okay, so we're doing things slightly different. And so, for example, this Willette addition, step 14, instead of inoculation loop and plastic cup of alcohol, you need the metal incubation loop, which is this right here, the metal tube rack, the sternoflame, and a lighter. And so the kit we're using slightly deviates from the stock kit because I want you guys to do some more real microbiology using an actual flame and actual loop. And so it's really important before you start doing any labs to read the entire lab, look at the Willette additions, and then probably what you want to do is, right, you need to print this out, say step 14, okay, then go find step 14 and make an annotation on there yourself. The software where I made the annotations only allows me to do it in certain places. Otherwise, I would have just changed the text right up there. But I didn't have the capability of that, and so do that. And the other thing is, please print out the directions and follow the directions from the printed form, okay? Not with a laptop, not looking at a computer, because when you're working with microbes, the last thing you want to do is then go over to your computer and use a mouse or scroll up or down, because then you have a chance of contaminating your computer. In the microbiology lab, for the most part, you're never pulling out your phone, you're never using a lot of things. What you work with in the lab stays in the lab, okay? And so from that perspective, please, please, please print out your lab guide for every lab and follow the written directions. Now certainly, read it online and understand it online. There's different colors and things like that, but when it comes time to doing the lab and annotating it, use the hard copy. Okay, so we have our plates. I already just washed my hands, and so what I'm going to do is I'm going to go ahead and use this isopropanol to go ahead and do a little bit of disinfection of my tray. And so I like to put enough on there so that after I wipe it down, there's still some alcohol on there, which can then evaporate. Because right now, the 70% alcohol is actually working against microbes. So let that then dry, and then we'll be ready to go. Okay, so the next thing we need to do is essentially conceptualize what we're about to do, right? Make sure that you read your instructions, and what the instructions say to do is essentially what we're going to do is we're going to sample from 4 places. You're going to get your feet wet, so to speak, by inoculating 4 plates from 2 human locations and 2 environmental locations. And what you'll see in the Willette additions as well as the flow chart, which I'll show you, it's also in PDF. It's actually, now why not bring it up now? It's at the end of this lab where it actually shows you, for example, down here, one thing that I've changed is, for example, 1 plate will be soil, 1 plate will be some other for the environment that you choose, and then 2 that are human. Okay, so basically I am requiring you to do a soil. And the reason that I'm requiring you to do a soil sample is because there's a lot of microbes in the soil, and you're guaranteed to see some interesting stuff. Okay, sometimes when you swab from the human, for whatever reason, you don't get much growth, so that's kind of our super positive control. Okay, so what we're going to do is we're going to go ahead and label the plates. And so if we look at these plates, let's orient ourselves. So you already made them, so you have an idea. If we look at this particular plate right here, there's a little bit of condensation on the lid. This one has a whole lot of condensation on the lid. And so the condensation on the lid is why when we incubate them for 48 hours, we do not do them right side up, because if condensation forms even more, it can then end up falling down, dripping down onto your sample, which you don't want. And another thing I'll bring up right now is that for the purpose of instruction, I will often open up a plate and show you, but you don't want to be doing this because right now microbes can be falling onto the plate from the environment, all right? So this is just so that I can show you certain things. So in general, always keep them closed, and in general, when you can, keep them all upside down. And upside down means that the agar is on the top and the lid is on the bottom. And again, the reason we do that mainly is so that condensation does not drip down, land onto the surface of the plate, and spread out your bacteria. Because what you're going to be doing with these plates is you're going to try to be isolating individual colonies. So first thing we need to do is we need to label these. And so I'm going to go ahead and label soil. For my other environment, I'm going to go ahead and see what kind of microbes are on my phone. But you get to choose whichever one you want for that. Everybody's doing soil, but the other environment, choose whatever you want. Okay. Over here, I'm going to go ahead and swab my nose. And the other one, I'm going to go ahead and swab, let's say, behind the ear. Okay. And so what you'll notice is where I labeled them, I did not label them in huge letters. Okay. And I did it in one section. So basically, wherever I labeled this, that is going to be my quadrant 1. Okay? So that will be my quadrant 1, and that is where I am going to actually swab my sample. So it's kind of a marker. Now when I go outside or wherever and I'm swabbing, I know that where I'm going to put it is directly over the marking. Okay? So let's go ahead and do that. So first what we're going to do is go ahead and let's swab from my body first. And so I'm going to do my nose, ear, phone, and then soil. And so what we need then are some of these swabs, okay? And as the instructions say, feel of them. This is the hard part right here, and this is the soft, swabby part, okay? So definitely this is not the part you want to open. And so for these, open them up like this, and only pull out one at a time, okay? And so you're only opening it just a little bit so that the swab that's still in there stays, in this case, the swab in there stays inside there so that when I pull this out, right, this other swab I can just put to the side and then I've got this that's good to go. Okay, so what I'm going to do is I'm going to go ahead and put this in the distilled water that I just poured. And the reason we're using distilled water is because there's going to be very, very low microbial load so that ideally whatever you see on your plates came from where you're sampling. The purpose of the water is just to get this swab moistened, right? So that's what we want. We want a nice moist swab. So now if we go to my nose then, what I'm going to do is I'm going to go ahead and just put it up in there and I'm going to gently roll it around, okay, on the inside. And I'm doing it multiple times because of whatever microbes are in there, I want to see, right? And there's a good chance that you will get a high number of microbes. And so don't just do it a little bit. The point is you want to see the microbes. Don't put it in there for a few seconds and be like, oh, well, I don't want to seem like I'm dirty. Everybody's got microbes in their nose, and the point of this class is for you to understand that and to, well, have some joyous experiences with your bacteria. Okay, so now that I've got my swab squared away, again, what I'm going to do is I'm going to put it on quadrant 1, which is right under the black marker. So there's two ways you can do this, okay? You can either do it like the lab manual says, where essentially you learn to keep your plate covered at all times so that if bacteria are falling down, it does not get in there. And in that case, what you want to do is essentially swab, okay, this first quadrant back and forth, back and forth, back and forth, right? And you don't want to go much further than where you marked it. And I'm going to go ahead and turn the swab over 180 degrees so that I'm making sure that whatever microbes I got on the swab come off. And the problem with this is that there's still going to be a lot of microbes on the swab because the swab has high surface area, microbes are going to stick to it. And so what we're trying to do is we're trying to make those bacteria go off of there. Okay, so that's what we've got there. And so once you've got that squared away, go ahead and close this up. Put it upside down out of the way. And now what do we do with this swab? Okay, well, the first thing that we're going to do with this swab is essentially I'm going to go ahead and swap out. I'm going to pull out the next one and put this back in there. So we don't have to worry about this swab quite yet. Okay, and so then for the ear, I'm going to go ahead and again moisten the swab. And then for my ear, what I'm going to do is I'm going to go ahead and sample behind the ear, right? And so I'm twirling it around, whatever wonderful microbes are growing back there, I want, okay? Another consideration, whenever you're choosing the portion of your body, don't have it be a portion that you cleaned just recently. So I wouldn't want to do my hands because I washed them really well before I started, okay? So again, for the ear then, we could do it the same way, where we open up and we kind of have that little barrier, right? And essentially what we're doing then is moving it back and forth, twirling it around. And so that essentially is our quadrant 1. And remember, the point is you want to get it on there. Don't do this, for 5 minutes. Don't press down hard. You're just skating it along. You're pressing down, somewhat firm so that you're making contact and the bacteria will absorb to the Petri dish. But there we go. And so when we do this first quadrant, what we're doing is we are actually inoculating the plate. That's the inoculation step. We are taking microbes from the nose, from the ear, from wherever it is, and we're putting it directly onto the Petri dish, which is full of nutrients, right? And so that's the inoculation step. When we actually streak with the inoculation loop, that is the quadrant streaking. Okay, but let's get back to our plates here. And so for the phone, what I'm going to do is I'm going to go ahead and check out my phone here. And now think about the phone, right? Often people will talk into their phone, like if you talk into your phone like this, for example, or like this, then you may think, oh, well, I want the bacteria from my mouth. Well, there may not be much around there. What I like to do is, when I'm using my phone, how do I normally hold it? I'm doing this, right? So my thumbs are touching the surface right there. And so that's essentially what I'm going to do. I'm going to go ahead and swab this particular area right here because when I'm texting, that's where my fingers are. Now, if you recently wiped down your screen, right, with some isopropanol or something, well then, good for you for doing that, but there may not be as many microbes on there. Okay, so for my phone then, again, what you do is you make sure you don't open up the swab part. You open up the wooden stick part. You pull it out. And by this point, you should pretty much know exactly what we're going to do here. And again, the liquid here is so that your swab will then be moistened and will pull bacteria off of whatever you're swabbing. Okay? So, from that perspective, let's go ahead and see what my phone has. Okay, so I'm going back and forth. I'm swabbing it around. Just like you're cleaning it, you want to pull all the microbes off of the surface. And now you don't want your swab to dry, so don't do it too long. And so another way you can work with this is instead of the way I did it before, you can just do this really quick. Now that does open you up to perhaps more contamination, but sometimes, especially when you're just starting out, this is a whole lot easier to do than trying to hold the lid at the same time. Okay, so for the first set of experiments for this lab, you're getting used to it, and I understand that. So do what works best for you, okay? So now we have the phone squared away. And so the next thing, now this was just the phone. This can just go in the regular garbage, right? This didn't come from a human sample. It was just my phone. So I'm just going to go ahead and throw this in my, my garbage. And now what we're going to do is we're going to go ahead and do the soil. Now, if you were also doing a door handle or something, that's where you take your stuff that you're going to use and you go to wherever it is that you're going to swab from. So what we're going to do then is we're going to go ahead and take this. We're going to grab our swab, our soil, and our distilled water, and we're going to go ahead and head outside and get into some soil. So let's go do that. Okay, so now we're going to go ahead and sample from the soil. So we need to do just what we've done before. We need to go ahead and moisten the swab. And this is particularly important for this particular location right here because if you look at it, this is fairly dry, sandy material. Often what you'll want is kind of more dark, rich. But in this case, since it's fairly dry, I'm just going to go ahead and take a stick around here, trying to get down a little bit more where it's a little bit moister, because we want to see the microbes. And we know they're in there. It's just that there's going to be less where it's sandy. Okay, so Now I'm going to go ahead and put it in. All right, cover the swab, and then basically what we need to do is just like we did on the others, I'll tap off some of that soil where we've got quadrant 1, and I'm twirling this around. When we're done, it should look a little bit dirty because, well, we're putting dirt on it, right? And so that should be plenty. So I'm going to go ahead and cap this up, and let's head back in, and we'll go ahead and do a quadrant streak. So now we've inoculated 4 plates on quadrant 1 with the swab. So the next step is to actually do the streaking using the inoculating loop. So let's go ahead and take a moment and do that on the board together, and so you can practice on a piece of paper. So before we actually do a quadrant streak on a plate, we should practice on some paper. So go ahead and practice this with me. And what we're going to practice are quadrant streaks. So by the fact that we've got quadrant means that there's 4 components to it, right? And so if we have a Petri dish, which I'm drawing here, what we're going to do, for example, well, in general, you always put in quadrant 1, you kind of paint it, okay, which means that you go back and forth, back and forth, back and forth. Often, for example, in the first lab with Ubiquity of microorganisms, we're taking a swab, and so this is what we're doing with the swab right here. Other times we may be taking it from a broth or from a plate, but still, we always, in the first quadrant, put a lot down and smear it all around to try to get it relatively evenly distributed in that quadrant 1, okay? And then, for example, if you're using a loop, right, then you would sterilize that loop. In between every quadrant, sterilize your loop. In the first lab, Ubiquity of microorganisms, we do a swab for the first one. But still, before you ever use a loop, you need to sterilize it. So now I've got a loop. I sterilize it. I wait 10 to 15 seconds. And then I start in the previous quadrant, in this case, quadrant 1, and I start there because what I'm doing is I've got a sterile loop. I'm taking it, I'm physically dragging bacteria out of there and into this area that doesn't have bacteria on it yet. I then bring my loop all the way back into quadrant 1 again, and I bring it out, okay? And then after that, I don't go back in quadrant 1. I just try to go back and forth, because by going in 1, 2, 3, from the first inoculum, which is going to be the most intense amount of bacteria, is the highest concentration. Ideally, I brought enough out, and as we go along, every time I move, there's less and less bacteria, right? I like to use the example of if I had a paintbrush and I were to load it up and I were to just start walking around a huge building on the outside, there'd be a lot of paint to begin with. I'd walk around, and over time, there'd be less and less paint being put on the wall because we're running out. It's the same idea as this. Okay, so then you take your loop, you sterilize it, you wait 15 seconds, 10 to 15 seconds. You start here, you drag bacteria out, you go back in, you drag it out, and then what? Do you want to go back in? Maybe not. You just then go like this, right? Then you sterilize your loop, you wait 10 to 15 seconds, and you start in quadrant 3. You bring it out, bring it back in, out, there. And I got duped again. This yellow marker looks just like the green marker, so I'm going to put this away. So what we have is essentially quadrants 1, quadrant 2, quadrant 3, and quadrant 4. So let's go ahead and do that again, right? So let's keep practicing because you want to make sure that you're really good with this. Okay, so we've got the beginning. We'll start with quadrant 1. Put a lot of bacteria, keep doing it, whatever the sample is, from your nose, from a broth, from the soil, from your phone, whatever the case may be. Okay, so good. So that's quadrant 1. So then you take, if you have a loop, you sterilize it, and then essentially you go ahead and start in quadrant 1, you bring it out to quadrant 2, back into quadrant 1, out to quadrant 2, and then you stay in quadrant 2. That's quadrant 2. You sterilize your loop, you wait 10 to 15 seconds, and then you start where there's bacteria, you drag it out, you go back in, you drag it out, and now don't go back in because you want to dilute it to a small concentration. And then for the last one, what we're looking at is essentially quadrant 4, start where there's some, come out, and then just use up as much of the space as possible. Okay, and so that is quadrant before. And so practicing with me like this, it's pretty simple. But I tell you, after teaching this for 10 years, there's a lot of ways that students get this wrong, because once you start doing it, it's so foreign if you've never done it before, right, that you need practice. And so that's where doing it on paper. So let's do a couple of examples of ones that don't look good, right, ones that you want to avoid. Okay, so for example, this is typically an okay one. But then sometimes what people will do is they will essentially do something like that. And now that's bad, because you see how you're going back and forth over and over and over, whereas this, you have parallel lines. If you keep going back over and over and over again, essentially what you've got is you're not diluting it. Remember, the purpose of doing this is diluting the bacteria so that there's smaller and smaller concentration. And if you keep going back and forth, then you're not diluting it. Okay? Another thing that some students will do is they'll start like over here, and they'll go in. Well, you just started an area where there's no bacteria, so although you may end up getting the right answer because you do go back in, right, you still may get isolated colonies. You did it in kind of a goofy way that doesn't make sense, right? But things like that, at least you can overcome as long as you've got bacteria growing on there, okay? I've seen plates where students, even after practicing, they go ahead and, this is often the one that works best, but then I'll see, how the plates look, and essentially they look like something like this, where they didn't, I don't know what happened, right, but that is clearly wrong. Okay, so you don't want to keep going over each one. You always want to start not where there's no bacteria, but you want to start where there is some bacteria, and you don't want to just keep going over and over and over again. Now, there are slightly different variations on the theme, all of which work. So for example, if you started out here, right, and some people like to just do something like this. And that's okay, right? Similarly, some people do this. I would accept that. So all of that will work. It's not the way I do it, right? But you could do something like that, as long as what we see on the plate is that there's a high concentration, after it develops, after it grows, after it's incubated, high concentration to lower to lower to lower. And the proof ultimately is, what is the growth pattern on the plates after your bacteria incubate? And that is where, part of the points are built, and that is, I can look at a good quality image of a plate, and I can tell pretty much whether you did a good version, right? And this is still good, right? Or you did a bad version, right? Well, this part up here isn't good, actually, right? Going back and forth I meant this part is okay. So anyway, this is quadrant streaking, and the point, again, the point of quadrant streaking is what? That's right, it is to get single colonies, to dilute from a high concentration, make it lower, lower, and lower, so that ultimately what you've got is a plate where you have some individual colonies scattered around, and then you can study them. So that is the purpose of a quadrant streak, to isolate individual colonies, often from a mixture. When you're sampling from your body or from the environment, there's going to be a mixture of bacteria in that sample, in that swab, for example, and so you need to be able to separate them out. And that's a quadrant streak. Okay, now that we know how to do that, let's go ahead and get rolling. So the first thing we're going to do is light up our sterno, get a nice big flame on that, and make sure we let it get nice and large, okay? And now for your loop, okay, what you're going to do is you need to sterilize, first of all, since this is the first time using today, the whole thing going up. And so what you want to do is, you'll have a tendency to want to do this and move back and forth. That's very inefficient. So instead what you want to do is you want to actually angle it up like this. Okay, and you see how the end there, it's getting red hot? That will incinerate any bacteria or fungi or viruses that are on there. And then what you do is you slowly move the loop all the way down so that the entire braided part essentially gets red at one time or another. Okay, so now that we've sterilized it, a very important thing to do is be patient. Okay, so first of all, I'm talking to you to explain this, but you wouldn't want to be talking because right now there are microbes leaving my mouth and going all over my work area down here. Okay, but basically we need to wait 10 to 15 seconds for this loop to cool. Because if you take a super hot loop and you put it to your bacteria, then essentially what you'll see is you'll kill a whole bunch of bacteria, which you don't want. So after you wait the 15 seconds, and doesn't matter which one we start with, then it is time to go ahead and do the quadrant streaking. So again, just like before, you can do it one of two ways. You can hold it right side up like this and work like this, where you're using the lid as a barrier. So I'll do that a couple times, and then I'll do it the other way. Okay? So the key thing is essentially to take it from quadrant 1, just like we showed on the board, right? And essentially bring it out into quadrant 2, bring it back into quadrant 1, out into quadrant 2, and then basically I'm not going to go back into quadrant 1, I'm going to stay in quadrant 2, and there we go. Okay? So if we look at this, it's hard to see here, but if you were to look at yours, or there we go, you see the streaks? There we go. We can see actually the streak marks right there. Because basically what we're doing is we're carving a little bit of the media, but not so hard that it breaks, but you can see those streak marks. Okay, so in between each streak, it is important to go ahead and sterilize the loop. Because by streaking, what you did was you put a bunch of bacteria on the loop in a very concentrated region, or we hope so. Okay, and so basically what we need to do then is wait 10 to 15 seconds for this to cool down before we take a streak from quadrant 2 and put it into quadrant 3. Okay, so we'll go ahead and get that going. And while it's cooling, I'm going to go ahead and get my bearings, make sure that I can see right where that quadrant was. Okay, and so for me, I can see it. It's kind of hard with these lights in here. There we go. Okay, so I can see, for example, that right there, what we've got is, this is quadrant 1. Okay, there we go. There's the streaks. Bear with me while I try to get it in the camera, right? And so basically what we have here is going from this quadrant into the next one, back in, and then out, and then back, and I'm not going in now. Okay? So the next thing to do is use the loop, sterilize it, let it cool, and now we're going to be doing the 4th quadrant as soon as this is cool enough. So I'm just looking to see if I can see my quadrants, and so there you can see the different areas that have been already struck. Again, patience is key. Don't start going right away, right? Okay, now that this is cool enough, I'm going to go ahead and go from quadrant 3 into quadrant 4, back into quadrant 3, back into quadrant 4, and now I'm just going to stay in quadrant 4. So the key thing is when I'm doing this, I can see the streak marks. It's just hard for me to look at it and at the same time show it to you on the camera. So once you're done with that, I'm going to go ahead and take this. I'm going to put it outside of my tray. That way I know that I've already done it. And let's go ahead and do these next samples fairly quickly. Remember to always sterilize your loop in between each one. Okay. I'm going to go ahead and do the soil next. And again, wait the time. Don't feel antsy. Don't feel as though you need to get to streaking right away. Wait the time so that the loop cools and you don't kill the microbes that you're trying to grow. Okay, now this one I'm going to go ahead and do it like this so that hopefully it's a little bit easier to see. And basically, right, quadrant 1 into quadrant 2, back to quadrant 1, into quadrant 2, back to quadrant 1, into quadrant 2. Now I'm staying in quadrant 2. I'm not going back into quadrant 1. Okay, go ahead and sterilize the loop in between each and every one. Another way to test too, if it's cool, or to cool it down a little bit more quickly, is you can find an area of the media that you've not inoculated and you can just gently touch it with this, and the coolness of the media will help cool down the loop. Okay, you would only want to do that with the sterilized loop. Okay, so now I'm looking at my plate. I'm going to start in quadrant move over, and then essentially just keep doing the same thing. And now what I'm going to do on these last two is, just for the sake of showing you without waiting the 20 seconds in between each one, so this video isn't super long, I'm just going to go ahead and not do my extra sterilization, my super important sterilization of the loop in between each streak, simply to save time. Okay, but don't you do this, right? This is just to illustrate a little bit more quickly, so we're not sitting here for a really long period of time. Okay, so I find quadrant 1, which is right along here, right? There's quadrant 1. And so what I'm going to do is I'm going to go ahead and streak from quadrant 1 into quadrant 2, back into quadrant 1, into quadrant 2, back into quadrant 1. Now I'm back into quadrant 2, and now I'm staying in quadrant 2 a couple times. Okay. And then you would flame, but I'm not going to right now. You would wait 20 seconds, and then you would go and start from quadrant 2 into quadrant 3, 2, and then I'm just going to stay in 3. Okay? And then what I would do is I would flame, and then I would go from quadrant 3 to quadrant 4, and basically I'm just going to go ahead and stay in that. And so what I'm going to do is I'm going to sterilize this loop before I put it away. And what you can see from this is essentially the different streak marks. Okay, so it is a little bit hard to see, but you can at least see them carved out. And I wonder if it'll be a little easier this way. That sure is flashing, isn't it? There we go. Okay, so from quadrant 1 down here, right? It then went into quadrant 2 over here, and then brought it into quadrant 3 over there. And quadrant 4 is that little squiggle right there. Okay, so hard to see, but on your plate you should definitely be able to see it. And what you want to do is, whatever plate you're using, when you're done, go ahead and look at it, right? It's hard for me to show you in the camera, but look at it to see if it is the same streaking pattern, right, as we did on the board together, which we're showing right here again, so that you can see what your pattern should look like. It's really important that after each time you do this, you look at it and then determine if it's a good streak, and if not, what do you need to do for the next plate? So every plate, ideally, should get better, right? So you're paying attention to what you're doing, you're learning from that, you're comparing it to your practice when you did it with me on the board, and then you're moving to the next one. Now, the last thing I want to mention, and then there's the fourth plate, which you would do, but I think I've shown you how to do it, so we don't need to belabor the point, right? But quadrant streaking is super important, because quadrant streaking is the method to isolate single colonies of a particular bacterium. And if you're going to study the bacterium, you need to be able to isolate individual colonies so that there's no contamination. So this is a fundamental technique of microbiologists, and that's why it is one of the first techniques you're learning. And now the other thing that I need to point out is, if you look around, you look at me, I don't have my safety goggles on, I don't have my apron on, I'm not wearing gloves, right? So what gives? Well, this first exercise, we are not working with high levels of microbes at all, right? We just sampled from ourselves, we sampled from the soil, and we struck. And so this is like one of the only times in lab where we don't need to be suited up to do microbiology labs. So what you'll see is, in the next set of labs, I will have all of my safety gear on for the lab, and that will allow me to do things safely. So that is the only reason why in the first lab you don't need to wear your goggles, you don't need to wear your gloves, you don't need to wear your aprons. That is because none of the plates we have right now have bacteria that we have grown. Because once they've grown on the plates and they're in high numbers, that is when there's a potential for contamination and for you to get sick. Okay, so now the last thing we need to do for this, after you've done all four, right, is essentially, you don't want that to happen, you don't want your plates to open up, is we then need to go ahead and put it in our incubation chamber. And what I'm doing is I am doing these essentially so that they are all agar side up, okay? And then I'm closing it up. And then the lab tells you to go ahead and, wherever you're going to put it, Make sure that the temperature is between 21 and 25 degrees Celsius, which is one of the reasons why you've got this thermometer. So what I do is I just put it on top, I make sure that it's between that, and actually if it gets up to 30 degrees Celsius, that's even better, okay? The reason why they don't want you to do it higher than 25 typically is because if you were to just put these plates somewhere higher than 25, the plates will start dehydrating. But if you've got it in a container like this, you could go as high as 30 degrees and still within 48 hours your plates will not dehydrate if you made them correctly. So let's go ahead and put this in some safe spot in your house, right, where you need to go. And then in 48 hours we will be back to look at the results on that. And as far as cleanup today, actually since all of these, this swab for my nose is just like if I blew my nose, and every time we blow our nose we just throw that in the garbage. So I can throw these in the regular garbage, right? Again, this is a lab where we haven't grown anything up yet, so it's a little bit different. However, one thing I do like to do just as a course of practice is every time I'm done with my tray to make sure that I go ahead and clean it. And in this case, we don't need the Clorox bleach because again, we did not use bacteria that we grew on the plates. So I'm just going to go ahead and just because I always like to make sure I clean up when I'm done, I'm going to go ahead and clean this up with isopropanol. And then after I accumulate everything and put it away, I'm going to go ahead and clean this whole surface all around here. You know, it might be your kitchen counter, it might be a table, wherever it is. Make sure that you clean it appropriately at the end every time. And going back to the video where when we were looking at the Clorox bottle, non-porous hard surface, no matter when you're doing microbiology, that is what you need. Hard, non-porous, hard and non-porous. So make sure that whatever surface you're using, it works. And if you're going to use a tray, don't use one that's got a bunch of ridges, like a roasting pan or something like that, because anytime you have other ridges, it's harder to clean up. Here we can just wipe it down really nice, and it cleans it very easily. Okay, so let's go ahead and wait 48 hours, and we'll be back to look at our plates.